First, I want to start by saying thank you so much for inviting me to speak today. I'm really excited to be here and to be presenting on this research that I conducted as part of my PhD while I was in the environmental engineering department at the University of Connecticut. A paper on this topic, actually, that I'll be covering today was recently published in Water Resources Research. And I included the link at the bottom because there's a lot more details in there on this study that I won't be able to get to today, but you're more than welcome to take a look at that or email me, and I can send you a copy if you aren't able to get access through the link. I want to start by thanking my PhD advisors. I worked with Christine Kurchak, Wei-Ling Wang, and Penny Vlahos at the University of Connecticut Environmental Engineering and Marine Sciences departments on this project. And it's really near and dear to me because it's the bulk of my dissertation research. The topic is future projections of water temperature and thermal stratification in Connecticut reservoirs and possible implications for cyanobacteria. The motivation for this work really came out of conversations I had with the regional water authority down in South Central Connecticut early on in my research endeavor, and they were concerned about how climate warming might impact water quality in their drinking water reservoirs. In particular, they had concerns with cyanobacteria in a few of them and eutrophication and how that might increase the likelihood of, potentially more toxic cyanobacteria blooms in the future, or higher algal growth in general, or the release of other things from sediments like manganese and phosphorus that make water treating drinking water more difficult or costly. And so that's just a little bit of background that I wanted to share before I get started. A lot of research has already been conducted on climate warming and cyanobacteria. I want to start by summarizing some of that here. As many of you, I'm sure, already know, nutrient availability, temperature, lake mixing, they're all really important parameters controlling cyanobacteria productivity. I included this figure on the right to show that the distribution of cyanobacteria or algae in the water column is really dependent on stratification and the amount of mixing that's going on in a lake or reservoir. So a strongly mixed lake or reservoir may have a more even distribution of cells per milliliter, like on the left side, and then a highly stratified lake or reservoir would be more likely to have a surface scum and a concentrated amount of cyanobacteria at the surface, and that's typically what we call a bloom there. And that condition happens in the summer in a lot of lakes when they're highly stratified and when wind mixing is low. And so what I'm really going to be diving into here is how long-term changes in water temperature and lake mixing might impact cyanobacteria productivity. And the bulk of this work really looks at future projections of water temperature and stratification in the study lakes that I assessed here. And a little bit more context about the background is that air temperature in the broader Northeast region is really increasing faster than any other region in the U.S. And so this is, a really timely issue here to be thinking about. And the warming climate, warmer water, stratified conditions provide a competitive advantage for resources and the ability to outcompete other algal species. For cyanobacteria, they have gas vesicles which allow them to move up and down in the water column and have access to sunlight, or during the day and then nutrients at night. So that ability in itself allows a lot of cyanobacteria species to outcompete other algal species in warm conditions. In addition, many cyanobacteria species have a competitive advantage in higher temperatures because their optimum growth rates are reached at those higher temperatures. And so I just wanted to provide that background and context before I dive into my specific research question, which is how might future changes in lake water temperature and stratification influence the frequency and severity of cyanobacteria blooms in Connecticut in particular. But that being said, the results from this are likely generalizable to other temperate regions, in the Northeast and other areas in the world. So this is the study area where the 6 lakes are located in south central Connecticut. And I included this to show that the watershed area surrounding each of these 6 lakes varies drastically. And 2 lakes in particular, Lake Whitney in the center and Lake Salt and Stall, they have a high amount of development in their watersheds. And that's relevant because it provides nutrient inputs into those lakes. And that background information is important when thinking about cyanobacteria productivity. Although this study doesn't really dig into nutrients, it's important to know, whether or not nutrients are available and in what amount in each of these lakes. And so this helps to show that Lake Whitney and Salt and Stall in particular, they do have higher amounts of nutrient input. So what I did here, and I'm not going to provide too much detail on the methodology because we don't have time to get into it, but I looked at long-term changes in water temperature, stratification, and cyanobacteria biomass. And the indicator for cyanobacteria biomass was actually phycocyanin fluorescence in cells per milliliter. And I had a depth profile measurement for each month from April to October from 2003 to 2018. So using that background data, that observational data, I was able to create a model that ended up in the end allowing me to predict water temperature and stratification from downscaled air temperature projection. And I was able to also use the observed relationship between temperature, stratification, and phycocyanin fluorescence as the indicator for cyanobacteria to say, what conditions blooms were more likely to happen in the past based on the historical data. In addition to that depth profile probe data, I had access to raw water grab samples that were taken on the same day. At about 5 meters depth. And so that it was analyzed using optical microscopy and the total algal cell counts and dominant algal species cell counts were derived from that. And just quickly, I want to say that the statistical downscaled air temperature data is taken, at the 4-kilometer resolution and downscaled from the GridMet data set, which is an observational air temperature data set, and that is what was used to predict future changes in surface temperature and stratification in the lake. In addition to that, I have future and historical global climate model air temperature projections that are used to look at how temperature and lake stratification might change in the future up to 2050. And those results are based on the average of 3 general circulation models under representative concentration pathway 8.5, which is the business as usual scenario. And the global circulation models chosen were best representative of the Connecticut region that we're in. And so we did a special analysis on that to determine which ones to use. the last thing I want to say about methodology is that quantifying cyanobacteria health risk levels was one difficult piece of this. And using the cyanan fluorescence depth profile data, I had to think about how to quantify that. And so in the end, what I ended up doing was taking the summation of the depth profile data that was taken at 1 meter depth interval, and using that total amount in cells per milliliter as the number that I compared against the low, moderate, and high, and very high relative probability of acute health effects levels for cyanobacteria risk. And so anything over 100,000 cells per milliliter would be high relative probability of acute health effects. But that being said, additional research is needed to understand the relationship between cyanobacteria biomass, cell counts, and cyanotoxin production. And I think Ingrid's presentation led well into that topic here, because just because there might be a high amount of cyanobacteria cells per milliliter, that doesn't necessarily mean that there are toxins in the reservoir. I'm going to, for sake of time, just skip through the rest of the methodology and say, if you're interested in that, I can talk to you about it afterwards, or we can, you can ask me questions at the end. But I want to just briefly go through the main points and the main findings of the study. So based on the observational data in the reservoirs that have had cyanobacteria blooms in the past, I found a strong relationship between total relative thermal resistance to mixing, which is a measure of stratification, and surface water temperature. This figure shows that relationship. The size of the circles here is the total cyanobacteria biomass in cells per milliliter. And so the big kind of take home here is that we start to see higher amounts of total cyanobacteria biomass, greater than 100,000 cells per milliliter, which is an indicator of high risk at temperatures that are above about 22 degrees to 23 degrees Celsius, and total RTRM above 250 in this particular case. And total RTRM is lake specific, so that number is not really generalizable across lakes at this time, but it's useful for understanding the relationship within a single lake. And this was done for each of the lakes, and we did see a relationship. I did see a relationship in the other lakes that also had cyanobacteria blooms. But lakes that don't have blooms, it's harder, of course, to determine the relationship between stratification and water temperature in those particular lakes, because the data was just not available. But in general, looking across the 6 lakes, we found, I found that shallower lakes with higher surface areas located in more developed areas are generally more vulnerable to increasing cyanobacteria blooms. In addition, laboratory samples showed that common temperate region cyanobacteria species capable of producing toxins did appear as dominant species in the samples taken. And these were the samples that were taken at times when the cyanobacteria biomass amount based on the phycocyanin fluorescence was above 100,000 cells per milliliter. And so some of the species that were present are just listed here. I'm not going to go through them now, but many of them are capable of producing microcystin, spermatoxin, toxin, and anatoxin. But again, I don't have data on those toxins, but the fact that they can produce them is reason enough to start monitoring in this particular example. And this is the final figure that I'm going to show, and some of the main findings of this study. And this figure here shows that from January to December, surface water temperature, average water temperature, bottom water temperature, total RTRM and max RTRM are increasing across all of the reservoirs. And so what data you see here is actually averaged across the 6 reservoirs that were assessed for this study. And the red line is past, the average of past temperatures for each month averaged from 1971 to 2000. And the blue line is the average temperatures for each month averaged for the future period of 2040 to 2070, using the future climate model projections that I mentioned a minute ago under the emissions scenario, RPC 8.5. And so just looking at the top left chart, for example, it shows that surface water temperatures is likely, is increasing across the year throughout all of the months. That blue line is above the red line for each month from January to December. And that's true for surface average and bottom water temperature based on the projections here. Stratification, which is measured based on total and maximum relative thermal resistance to mixing on the right side here, that shows that the bigger change there is happening from April to to November. And the largest increase in stratification is really expected from about July to September. So, to conclude, I have a few slides with the take-home points. Higher annual air temperatures are driving higher annual surface water temperatures and higher total RTRM. And Connecticut lakes are becoming warmer across the year and more stratified during summer. On average, annual surface and bottom water temperature will continue to increase significantly per decade, and extreme cold and moderately high surface temperatures are projected to decrease while extreme hot temperatures go up. And I don't have there's another figure and table that gets into that in the paper, but I wanted to just make that point here. And just to put it into perspective, it's really about 20, we'll have about 20 less days with temperatures in the 20 to 25 degree Celsius range by 2050. And an additional about 50 days over 25 degrees C by 2050. There's I saw little variability across lakes, and the results are similar for surface average and bottom. There are again significant increases in stratification, likely from July to September, and stratification is likely to begin about 2 to 4 weeks earlier and last 2 to 4 weeks longer by mid-century. And I will just end here with the key points relevant to cyanobacteria, and that's that lake conditions are changing, and the future conditions are likely to favor cyanobacteria over other algal species more often in the future. And that's especially in lakes that have a history of cyanobacteria blooms. And I will just end there because I know we're probably out of time. But any questions?